Hello, everyone, and welcome to today's webinar, Advancing Cell Biology with Cryo Correlative Microscopy. I am Jennifer Woods of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Microsystems and Alvéole. To learn more, visit leicamicrosystems.com and alveolelab.com. Let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Elizabeth Wright, Ph.D., Professor of Biochemistry, University of Wisconsin-Madison, Morgridge Institute for Research, Jae E, Ph.D., and Bryan Sibert, Ph.D., Postdoctoral Research Associates in the Wright Laboratory, and Joe Kim, Graduate Student in the Wright Laboratory. For a complete biography on our speakers, please visit the Biography tab at the top of your screen. Dr. Wright, you may now begin your presentation.
Good afternoon. Thank you for joining us for today's webinar. This is the last webinar of our three-part series on Cryo-CLEM. During today's webinar, we will discuss how correlative cryo-microscopy is advancing cell biology. We would like to thank our sponsors and co-organizers, Leica Microsystems and Alvéole Labs. We would also like to thank LabRoots for hosting this webinar series. As mentioned, my name is Elizabeth Wright. I am a professor at the University of Wisconsin-Madison. Our research group focuses on questions related to infectious disease and human cell dysfunction. We also develop and use a range of cryo-correlative imaging methods in our research. I would like to introduce three other presenters for this webinar and the extended webinar series. Dr. Jae E is a cryo-EM biophysicist in the group and develops light microscopy, cryo-EM, and cryo-CLEM methods and software tools. Dr. Bryan Sibert is a structural virologist on the team. He develops and uses light microscopy, cryo-EM, and cryo-CLEM workflows for virology research. Mr. Joseph Kim is a Ph.D. student in physical chemistry at UW-Madison. He develops and uses light microscopy, cryo-EM, and cryo-CLEM workflows for neuroscience research. If you missed our first two webinars or would like a refresher on their contents in the future, you may access the material at LabRoots. The first webinar, Correlative Cryoimaging for Cell Biology, was presented on June 11, 2020. In that webinar, we introduced Cryo-EM at UW-Madison, highlighted major emphasis areas in structural biology and connections to Cryo-EM and cryo-CLEM, and we discussed some of the theoretical and practical aspects of labeling biological targets and included research examples. We also discussed CLEM equipment options and workflows, including research examples. I presented some areas for CLEM technology development and a few suggestions about resources. In the second webinar, a hands-on tutorial, Workflow Advances for Whole Cell Correlative Cryo-Microscopy. This was presented on July 2nd, 2020. In that webinar, we discussed Cryo-EM at UW-Madison and highlighted aspects of Cryo-EM and CLEM. We considered best practices for cell culture, micropatterning workflows, and advanced cryo-CLEM strategies. Let's begin here with a brief overview of today's webinar. I will introduce Cryo-EM at UW-Madison and mention some key reasons for why cryo and why CLEM. From there, I will discuss Cryo-CLEM equipment options and workflows. Then we shift to a practical discussion of micropatterning with the Alvéole PRIMO system and Cryo-CLEM with the Leica EM Cryo-CLEM. During each of these portions, I will highlight some biological results. I will present some future areas for CLEM technology development. I will then provide a few suggestions about resources, make a few acknowledgments, and point to our previous webinar events. The Wright Lab moved to UW-Madison in 2018 to begin our next phase of work in cryo-EM structural studies of bacteria, human cells, and viruses. Simultaneously, we set out to build a cryo-EM research center at UW-Madison in the Department of Biochemistry to support the research endeavors of investigators across the UW-Madison campus, regionally, nationally, and internationally. We have assembled a team of the PI, 2 staff scientists, Eric and Matt, 4 postdoctoral research associates, Jae, Bryan, Julene, and Daniel, and 3 graduate students, Joe, Juan, and Vicky. Members of the group are engaged in facility management and technology development, research projects in the areas of infectious disease and neuroscience, and cryo-EM education. We are always on the lookout for new team members. In addition to our team, we have a suite of instrumentation to support all major cryo-EM workflows, from the basics of sample optimization by negative stain TEM to single particle data collection and 3D map determination. We specialize in areas associated with cryo-CLEM and cryo-electron tomography and have built resources to support studies of biological targets from light level imaging through cryo-FIB, SEM sample milling, and cryo-ET data collection, 3D reconstruction, rendering, and subvolume averaging. The members of the team and instrumentation are essential for the success of cryo-EM studies in the Wright Lab and the Cryo-EM Research Center at UW-Madison. Recently, the NIH funded 4 institutions to build a national network of cryo-electron tomography centers. UW-Madison will be the central cryo-ET hub to support specimen preparation, data collection, data processing, and coordination with the 3 other centers. Specimen preparation service centers will be located at CU Boulder, the New York Structural Biology Center, and Stanford SLAC. We anticipate rolling out services in late 2021 or early 2022. Now, back to cryo-CLEM. Why are researchers actively developing strategies that use fluorescence microscopy, correlative light and electron microscopy, and cryo-EM? Briefly, cryo-EM technologies were pioneered to retain specimen hydration and reduce electron beam radiation damage to the biological sample while imaged in the vacuum of the electron microscope. These advances allowed data to be collected that could be computationally processed to generate native 3D structures of macromolecules from nanometer to atomic resolution levels. In kind, fluorescence microscopy was developed for resolving targets of interest during the imaging of living or fixed specimens. Finally, correlative light and electron microscopy, or CLEM technologies and methods, grew from the need to define the 3D structures of macromolecular complexes by EM that had been spatially resolved in biological specimens by light microscopy. In cryo-CLEM, the goal is to achieve direct correlation data from the specimen. As we consider the cryo-EM workflow to use, so much depends really on the biological question. What is our target? An isolated macromolecule or macromolecular complex, whole cells, perturbed cells, or tissue? And simultaneous to these considerations of the biological target, selection of the technologies used for sample preservation and imaging will also impact the relationship between context and achievable resolution. When we move forward with a research project, we consider the workflow in 4 phases. The first is sample optimization and preparation. The second is specimen preservation. The third is the imaging pathway. and the fourth is the computation and data analysis. Within this series of webinars, we have focused on the central pipeline for imaging intact cells for light microscopy, cryofluorescence microscopy, and cryo-electron tomography. Truly, everyone is investigating exciting biological questions that may benefit from light and electron microscopy imaging. However, we may not know what resources we need for addressing these questions and whether we should house them in our own laboratories or use them as part of a shared resource. There are many options available for equipment, and decisions regarding the systems to purchase and use depend on your experimental factors that are generally determined during the initial development of a project and refined over the course of the investigation. For the ideal and highly automated cryo-CLEM workflow, one would need access to live cell imaging systems, a plunge freezer, a cryo-CLEM microscope, a cryo-FIB SEM for sample thinning, and a cryo-TEM. So how do we practically do cryo-CLEM? In our lab and other labs, we have developed a cryo-CLEM workflow that includes standard operating procedures in areas that may require optimization to improve the success rate of cryo-CLEM experiments for the novice and the experienced practitioner. This workflow is conveniently color-coded to help the investigator work through each step and the associated steps of the process. Two areas within this workflow are called out with red and blue boxes for two reasons. First, when working with a variety of cell cultures, both eukaryotic and prokaryotic, it is critical to maintain a sterile working environment using the appropriate environmental controls and to use aseptic or sterile techniques to eliminate possible sources of sample contamination. Second, once samples have been plunge frozen, it is essential to maintain the samples, tools, and equipment at temperatures much lower than -150 degrees Celsius to minimize specimen devitrification or contamination. In order to help individuals plan and troubleshoot whole cell cryo-CLEM experiments, I will outline a few steps within the workflow. During the process, when we transition to grid preparation, there are a number of considerations. Briefly, each member of your team should have a standard set of tools and laboratory equipment to minimize specimen contamination and maintain your continuity of research. We prefer to use matte tech dishes for CLEM imaging experiments because they have a glass cover slip in the bottom that allows for periodic light microscopy imaging without the introduction of additional grid handling steps. We commonly add an additional thin layer of carbon to the grid surface to improve substrate stability and increase cell happiness. Depending on the attributes of the biological target, we may use perforated or continuous carbon films. One of the most critical steps is that we may also add an extracellular matrix protein across the grid to enhance cell adhesion and spreading. At this step, we may define regions of the grid where the extracellular matrix material is applied and encourage ordered cell adhesion and spreading. Finally, we add both fluorescent and electron-dense markers to the sample to support correlation and image alignment during the reconstruction process. An area of development in our lab and several other labs in the cryo-EM community is micropatterning to support complex cell culturing experiments and the improved automation through the CLEM pipeline. The leading rationale for micropatterning is that cells, be they eukaryotic or prokaryotic, really do not grow in two dimensions or in isolation. Therefore, technologies can be developed to support the expansion of the native in situ microenvironments on a substrate. This will increase our capacity to understand fundamental cell biology and design experiments to tease out architectural, mechanical, physiological, and other structural and functional relationships present between cells in a tissue and organ systems. In this schematic from Alvéole Labs, we review the basic steps in the maskless photopatterning process. First, the substrate, such as an EM grid or light microscopy cover slip, is coated with an anti-fouling agent. And then the photoactivatable PLPP liquid or gel agent. Second, the user will prepare a pattern with a basic image creation software. The pattern can be simply a rectangular pattern of ordered lines to complex patterns that include multiple geometries and possible gradients within the pattern. The pattern is then transferred to the Leonardo software that will control the substrate patterning process on the microscope. Third, the substrate is loaded onto the microscope platform, registered, and then with UV illumination, the pattern is introduced onto the coated substrate. The illumination process causes local degradation of the PEG polymer. This material and the excess PLPP agent is then rinsed away. In the fourth step, the user applies an extracellular matrix material, antibodies, or other biological agent. This material will remain only where the patterned process occurred. Finally, one is able to seed cells at an appropriate density onto the patterned substrate. At this point, we recommend visiting the Alvéole Lab website to watch a complete video tutorial on TEM grid micropatterning. In addition, there are links to several papers from the Cryo-EM community on the use of micropatterning in cell biology. Also, please really reach out to the Alvéole Lab team and the growing user community. Bryan and Joe from my lab are happy to share our experience. We hope you take the time to visit the Alvéole Lab website for more information. In Bryan and Joe's experience, the most critical steps to consider for the cells are the design of the pattern and the selection of the ECM material. It is important to keep the cell type in mind, in particular, what is known about their native environment. For the patterning process, use a stencil to maintain grid support and hydration on the cover slip or MatTek dish. In addition, during the incubation periods, keep the grids hydrated in a light protective container. Finally, how long does the process take? In our hands, we are able to move through the pattern design to cell seeding on 8 grids in a matter of 5 hours. Bryan has also been able to prepare a batch of pattern grids and store them for approximately a week prior to using them in a cell culture and imaging experiment. Other groups have reported longer storage to use times. Neurons have been imaged by a wide range of light and electron microscopy methods. Studies have been performed on cells isolated from tissue, cells in intact tissue slices, and cells of neural organoids. Microscopy studies of neural cells have advanced our understanding of basic cell function, cell development, neural cell communication, and a wide range of neurological diseases, including neurodegeneration. The basic morphology of a neuron is depicted in the cartoon. From left to right, we have the soma, which is the main body of the neuron that contains the nucleus. From the soma, smaller dendrites extend and branch off. The axon, or nerve fiber, extends from the soma and carries nerve impulses away from the cell body and to the next cell via the axon terminals that connect with the next cell's dendrites over the neurological synapse. Joe has been working with Tanner Tempas from Eric Dent's lab to culture mouse cortical neurons on EM grids. In the light microscopy image on the left, we can see several neurons, their soma, and the dendritic extensions. In the middle image, Joe has taken that grid, plunge frozen it, and imaged it under cryo conditions in the electron microscope. At this EM imaging level, we are able to visualize cell bodies, dendrites, and axons. We are also able to correlate light microscopy and EM level images to identify specific regions of the grid and target certain cells or locations on cells for higher resolution imaging. So, once you have mapped out your grid, selected specific regions of interest, and identified these cells of interest, it is time to collect tomography tilt series. A tilt series is a collection of 2D images that are iteratively acquired as the sample is tilted in the microscope. Images are captured at defined tilt angles while the sample is tilted. However, here we see a cryo tilt series that Joe has collected of a mouse cortical neuron. The grid is a metal mesh that supports a perforated carbon film layer. Ideally, one collects images over a hole in this carbon film. As the movie runs, we are able to see many features of the cell that will be further revealed upon reconstruction. After the tilt series is acquired, computational algorithms are used to generate a three-dimensional reconstruction or tomogram. In this movie, we are now slicing through the 3D volume of the cell, or an extension of this cell. As we look closely, we are able to identify cell membranes, a mitochondrion, microtubules, vesicles, and small macromolecules and macromolecular complexes. It is definitely a rich environment that supports our ability to define the inner workings of cells in order to better understand how they function and the impact of disease on the cellular steady state. While examining 2D images and 3D reconstructions of cells is very informative with those with experience, it is also valuable to render or segment cells in a manner that makes the information accessible for other researchers. Here, Joe has used the EMAN2 neural network tools to segment the major structural features of the neuron. In yellow, we have the mitochondrion. In light blue, the microtubules. The vesicles are in purple, and the cell membrane is gray. Viewing a segmentation in 2D or 3D space allows us to better investigate the internal architecture of cells and where organelles, macromolecules, and membranes come together to regulate cell function. As mentioned, neurons have been imaged by a wide range of light and electron microscopy methods. Recently, investigators have begun using micropatterning approaches with neurons. This has opened up a broad range of research options, including the capacity to direct cell growth, options for defining neuronal cell networks, and studies of network development, and others of neuron cell signaling. A beautiful example of neuron cell networks formed on a micropattern substrate comes from Nathalie Piette from the Studer Lab in Bordeaux. Here, the cells have organized in a pattern reminiscent of an extended computer circuit board. In a second project in the lab, Joe has been culturing neurons from other organisms, including fly larvae, on EM grids. This is a collaboration with the Wildonger Lab. By employing micropatterning, we are able to define where cells grow on the grid to support fluorescence microscopy and cryo-EM experiments, minimize their spreading across grid bars, and foster patterns that support cell spreading and forming cell-to-cell contacts. In the left-hand image, we have a light microscopy map of the entire grid. Here, in the blue channel, we are able to see the ECM matrix pattern that Joe has produced with the PRIMO system. It is a pattern of X's that do not extend outside of the carbon film areas on the grid. In the higher magnification blowups on the right-hand side, we are able to view individual neurons in green that have seeded onto the pattern, again in blue, adhered and spread only along the ECM coating of this pattern. Once the neurons were happily growing along the micropattern regions on the EM grids, Jae plunge froze the samples. From there, he imaged them by cryo-EM at multiple magnification levels. In the top middle panel, we see the entire grid as we did by light microscopy at the left, and are able to pinpoint and mark specific grid squares with pattern neurons for closer inspection. These are the green squares. In the upper right-hand panel, we identified a cell, body, or soma in a single grid square and selected portions of dendrites for higher magnification imaging. One of those regions is shown in the lower panel. Here, Jae was able to identify a number of vesicular structures and ordered highways of microtubules. They are bound by the blue stars. Further analyses are underway to learn more about the overall architecture of these cells and the organelles and macromolecules within them. As you have seen in the previous webinars, we have a robust workflow for cryo-CLEM studies of cells and virus-infected cells. This workflow relies strongly on tools from Alvéole labs, i.e. the PRIMO system, and Leica, namely the EMGP and the EM Cryo-CLEM. With these tools in hand, we are developing improved routines for better automation and correlation between cryofluorescence microscopy and the cryo-EM data. Here, Jae demonstrates the steps of the basic workflow for correlation between cryofluorescence microscopy from the Leica Cryo-CLEM and cryo-EM data from a 300 kV side entry microscope. The data is of an RSV-infected cell, where virus replication competent cells are indicated in red, and the native immunolabeled virus particles fluoresce in green. So, now that you have prepared your sample, imaged it by light microscopy, and vitrified it via plunge freezing or high pressure freezing, it is time for cryofluorescence microscopy imaging. There are many instrument options available. The selection of the system depends on the attributes of the particular system as well as your biological targets. We decided upon a system that is part of a seamless workflow, has minimal transfer steps between the systems, and methods for handling that eliminate possible sample damage, as well as that the imaging and coordinate data is readily correlatable to the electron microscope. For our work, we have had great success with the Leica EM Cryo-CLEM system for studies of bacteria, human cells, and virus-infected cells. The Leica Cryo-CLEM has 11 major components. 1. The liquid nitrogen pump for cooling the cryo stage. 2. The temperature control unit that regulates pump function and monitors system temperature. 3. The DM6 microscope. 4. A 50x thermally isolated cryo objective. 5. The cryo-CLEM stage that supports loading and imaging of 2 samples. 6. The overall microscope control unit. 7. The stage control unit. 8. The cryo transfer shuttle used for loading grids into the chuck and onto the cryo-CLEM stage. 9. The fluorescent light source. 10. A CMOS or other type of camera. And 11. The PC with the LASX and other software components. So, you have prepared your samples, loaded them into the cryo-CLEM transfer shuttle, and you are ready to begin CLEM imaging. In this and the next slide, we will review the steps associated with imaging a single PRIMO micropatterned cryo-EM grid on the Leica Cryo-CLEM. The entire process per grid, depending on grid quality, target number of regions of interest, the number of fluorescence channels needed may take up to 1.5 hours. Here we see Jae at the cryo-CLEM imaging cells. Again, we performed this experiment with a single PRIMO micropatterned cryo-EM grid. The cryo-CLEM imaging took 1.5 hours. On this grid, BAS2B cells were cultured and subsequently infected with RSV, i.e. respiratory syncytial virus. The video captures screenshots of the 21 steps of the grid mapping and imaging process in the LASX matrix screener. The first 10 steps take approximately 20 minutes and set up the basic imaging conditions for the grid. In step 1, the grid template and the grid are aligned for the user to select the imaging parameters such as filter cube sets, camera exposure times, and single or multiple channels per position. In steps 2 through 4, the user sets up the imaging parameters for focus and selects focus positions. In steps 5 and 6, the user runs the focus map at the pre-selected focus positions across the entire grid. Now in steps 7 through 10, the user often reruns, with some manual input, the focus routines at selected positions to fine-tune the parameters. The next 6 steps will set up mosaic acquisition and generally take about 10 minutes to complete. In step 11, the user defines the mosaic areas for data to be acquired. In steps 12 through 15, the imaging conditions for mosaics are refined and may include adjustments to the filter sets, exposure time, and Z-stack heights. In step 16, the export parameters are validated and adjusted if needed. During steps 17 through 19, the grid mosaic is acquired. This process may take up to 40 minutes depending on the number of channels used, height of the Z-stack, sampling steps used, and areas collected. The final 2 steps of the Cryo-CLEM imaging process are to identify and mark registration points over the entire grid, as seen in yellow. This is step 20. And then identify targets of interest for cryo-EM data collection, squares with the green points, and object of interest in the blue points. Step 21. Now, at last, you are finished. You may continue to the second grid or finish the process for a single grid. Save and export your data and recover your grid for transfer to the cryo-electron microscope. Because cryo-CLEM is complex and relies on the need for accurate and timely alignments and correlations to be made between fluorescence microscopy and electron microscopy data, we developed Correlator. Here is an overview of the cryo-CLEM workflow with Correlator and serial EM. This diagram showcases the integrated feedback-based alignment loop between Correlator and serial EM. The dashed gray boxes denote the main operations performed in Correlator. The solid gray boxes surround the main operations that are performed with serial EM. Also noted are areas of overlap in the serial EM Navigator File 1 and File 2 that link Correlator and serial EM. Purple arrows indicate the iterative flow of the user-in-the-loop alignment that is followed until accurate correlations are achieved. So you've made it again. You have loaded your sample into the Cryo-TEM. You have your maps and navigator files. Now what? Briefly, with 5 screen captures from serial EM on a Titan Krios, we will walk you through the major correlation steps on the previous PRIMO micro-patterned grid. The first step is to import the Cryo-CLEM mosaic, the image. Then you will import the navigator registration points that are noted in yellow. From there, you will transfer the Cryo-CLEM image to and the navigator points. This is followed by the transform process that will allow the image and the navigator points to transfer to the TEM map, the pink and red markers. Now that you have a large-scale map and the points are in register, you can begin to work on finer correlation processes at the level of the grid square, hole, and object of interest. In this cryo-TEM image, we can see the orientation of the BAS2B cell along the same trajectory as the PRIMO micropatterned area. We are excited to remind you that Correlator is readily adaptable for CLEM and cryo-CLEM imaging of virus-infected cells, mammalian cells, bacteria, or other systems. In this panel, we are demonstrating the multifaceted workflow as applied to RSV-infected cells. In panel B, in the upper left hand, we see the Cryo-EM grid map where a region of interest is noted with a green box. In C, we note the initial live cell fluorescence microscopy mosaic of the same grid as in B, with fluorescent signals from RSV-infected cells in red, the RSV-F glycoprotein on the RSV-infected cells or released particles in green, and cellular DNA with the Hoche stain in blue. In D, we illustrate a serial EM screenshot of the Cryo-TEM map of panel B after it was correlated with the live cell fluorescence microscopy map of C in Correlator. The maps for the green region of interest squares were identified and collected. Blue boxes note many regions of interest on the entire map. Panels E and G represent the full CLEM workflow that corresponds to the green region of interest in panel D, with a live cell imaging square in E, a processed cryofluorescence microscopy image in F, and a cryo-EM map at 470x of the region in G. In panels H and I, there are superimpositions of the correlated live cell fluorescence microscopy image, i.e. in E and H, and the processed cryofluorescence microscopy image that's noted in F and I, with the EM map in G respectively. The same regions of interest of RSV particles were present and identified in both fluorescence microscopy modalities, i.e. live cell imaging and cryo-CLEM imaging. These are noted with the matching colored boxes. Panel J presents a central region with a thickness of 9 nanometers through a tomogram of RSV collected at the red boxed region. As with all areas in structural biology, many of us are looking at the future of CLEM imaging. Are there improvements to the methods and technologies that will lead to better and faster results? Absolutely yes. We can consider a range of improvements. In particular, we are interested in improving substrates and cellular culturing methods through micropatterning and microfluidics. We are also engaged in the developing of improved automation routines through software and hardware advances. And as you have seen throughout the webinar series, we have a robust workflow for cryo-CLEM studies of cells and virus-infected cells. This workflow relies on tools from Alvéole Labs and Leica Microsystems, namely the DMI-8 microscope with the Alvéole PRIMO system, the EMGP, and the EM Cryo-CLEM. With these tools in hand, we are developing improved routines for cell culturing along with better automation and correlation between cryofluorescence microscopy and cryo-EM data. One always asks, what's next? How do I begin my project? And here's a list of resources within the community and from Leica and Alvéole Labs. There are numerous ways to connect with cryo-EM experts as a community. We are all very happy to answer your questions. I'd like to take a moment to acknowledge a few members of our team. Historically, cryo-CLEM developments in the group were led by Sherry Hampton, Josh Strauss, Rebecca Dillard, Zunlong Qi, and Hong Yi. Our latest advances at UW-Madison have been pioneered by the efforts of Jae E, Bryan Sibert, Joe Kim, and Matt Larson. We have also had wonderful support from our colleagues at Leica, Louise Bertrand and Nancy Rizzo, and at Alvéole Labs with Aurélien Dubois and Serge Kadura. Here, I have highlighted the previous webinars in the series. They are available for viewing at any time from the LabRoots website. In addition, webinars are also located on the Leica, Alvéole Labs, and Cryo-EM Lab websites. We very much thank you for your attention today and with the other webinars, and we will now pause for questions.
Thank you for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you would like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many questions as we have time for. Let's get started. Our first question is for Bryan. You mentioned in your presentation that you seed the cells in appropriate density onto the grid pattern substrate. Please, could you explain how this is controlled?
Great question. Before we seed the cells on the grid, we count them and dilute them to a target density. Depending on the experiment, we either add the cells to the grids all at once, or if we're using a micro-patterned grid, we add the cells just a few hundred cells at a time and monitor their adhesion to the patterns with a light microscope. And once all the patterns are occupied by cells, we stop adding cells and incubate the grids. This does need to be optimized for each cell type that you want to use.
Thank you, Bryan. Our next question here is for Joe. In cell clusters, the cell-to-cell connections and arrangement are critical to overall function based on their polarity. Can micro-patterning reproduce these connections?
It is entirely possible that such connections can be reproduced by micropatterning. There was a previous slide throughout the presentation where a pattern, while enabled on glass, enabled an entire neural circuit to be constructed through a synaptic network. As long as one takes certain consideration in the placement of specific regions of the cell in specific patterns and locations, it's believed that many cell-to-cell interactions can be patterned through micropatterning, while also controlling for polarity and distances between adjacent cells for a variety of different cell-to-cell connections and interactions.
All right, thank you, Joe. Our next question is for Jae. In slide 32, figure F, you mentioned this is a processed image of E showing more defined details. Please, could you explain what type of processing was done?
Good question. So actually, E and F are from different imaging modalities. E is a live cell fluorescent microscopy image prior to plant freezing, and it's visualized under oil immersion objective lens at room temperature. And F is a processed cryo-FLM image after the identical cells being plant frozen. So in our previous webinars and other Leica webinars, we've been showing that more details could be revealed after advanced image processing tools. And then so here, the technique we're using is actually called a small volume computational clearance implemented in the Leica Thunder package. And this processing actually allows for preservation of the pixel coordinate position and subsequent accurate alignment and correlative imaging. So we show E and F here just to demonstrate the flexibility and adaptability of cryo-CLEM workflow to other existing systems.
Thank you, Jae. Our next question here is for Elizabeth. Let me pull it up. Oh, I'm sorry. Okay. When you used grid box, is it possible that the important information located on the line region of the box is, if this has occurred, how do you solve that problem?
That's a great question, and I think what the person is asking is, when you have grids, if the sample is located on the grid bar, i.e. the metal mesh substrate. If not, please email me offline and I'll be happy to answer more completely. So typically we are not imaging through that area because the grid bar is a metal and the electron beam will not penetrate that area. Now, if you're using a combination of FIB-SEM technologies where you could image by scanning electron microscopy, you could reveal the surface topology of that particular cell or object of interest. Alternative methods are depending on how you prepare the grid and what type of grid substrate is there. You may use the FIB milling approaches or even in some cases ultramicrotomy where you could mill and remove that metal mesh to reveal some of the cell structure. That's a little bit more tricky, but it can be done.
Well, thank you, Elizabeth. Our next question here is for Bryan. Is grid preparation and micropatterning similar to what you have described if using yeast such as Saccharomyces cerevisiae?
That's a great question. We have not done any micropatterning with yeast, but if there is a matrix protein or other protein that the yeast will preferentially adhere to, then it should be applicable to yeast or any other cell type that has an extracellular matrix protein equivalent.
Thank you, Bryan. Okay, our next question here is also for Elizabeth. You use plunge freezing in your methods. Would there be an application of high pressure freezing for somewhat larger samples with tissues?
Again, a really fantastic question. And so, yes, absolutely. So for more conventional pipelines, so in the earlier section of the talk, we gave a workflow, and on the right-hand side, there were workflows where you would transition from high-pressure freezing to free-substituted and embedded specimens. You can also do Simovis processes or FIB milling processes on tissue-type specimens that would allow you to spin them down for imaging in the TEM if you're interested in doing vitreous work. And again, we're happy to answer more fully offline if you would like greater detail.
Thank you, Elizabeth. Our next question is for Joe. Just a general question. He says, many cells are micrometer thick. If we cannot switch to different cell types, is there any method or trick that would allow us to collect cryo-ET data in addition to FIB milling?
This is a fantastic question, actually. And in addition to FIB milling, as well as other sectioning processes such as ultramicrotomy, perhaps one consideration could be, depending on the biological project in mind, is there a potential morphological alteration that can occur that would allow the cell to become thin enough for direct imaging on the electron microscope, such as, say, as an example, an application of either a plasma transfection or a drug that could, say, induce lamellipodia-like phenotypes on that cell that would definitely be thin enough for electron imaging. So not only are there physical methods, but there could be creative biological methods that can induce direct imaging without, in addition to FIB milling if necessary.
Thank you so much. Okay, our next question here. Let's see, we've gone to that one. I lost my place, I'm sorry. Okay, this one is for Bryan. If a cryo-CLEM is not available, can micropatterning be used as a coordinate system for tracking cell locations between room temperature, fluorescence microscopy, and EM imaging sessions?
Yeah, that's a great question. And the answer is yes. It's actually something we do regularly. You can position the cells using micropatterning such that you can easily determine grid orientation in both the fluorescent and TEM map. But it's important to keep in mind that cells may be damaged or removed during the subsequent handling and freeze processing, so it won't be as similar as it is with cryo-CLEM.
Okay, thank you, Bryan. Our next question is for Jae. Great presentation, thank you. I would like to know where and how in the workflow you are planning to limit the transfer steps between vitrification systems and microscopes.
This is a good question because transferring is very risky. So the live cell FLM imaging, the workflow we show here is prior to plunge frozen, so we don't have the vitrification part to worry about. But the subsequent cryo-FLM workflow could be conducted on bare grids or clipped grids. So if you clip your grids, you will actually avoid some damage during the clipping and then do a more accurate correlation. And then right now, Leica auto grid shuttle actually can be loaded directly onto the ocular stage. And this can actually help minimize the ice contamination and devitrification or other grid handling process. So I think it's doable.
Thank you, Jae. Our next question is for Joe. For neurons or other long cells, can micropatterning reliably cross grid bars such that axons in one grid square and soma in a different grid square could be reliably known as a single cell?
This is a fantastic question. While we have not tested this yet, we believe our micropatterning protocol will allow longer cells to cross the grid bars. This will be especially important for cells with long extensions, such as mouse or rat neurons. To ensure that the axon in one grid square and the soma in a different grid square can be connected as a single cell, one option could be to create a buffer zone of unpatterned squares around the patterned squares themselves to make sure that no other cells can interfere with the designated area. Upright cryo-CLEM microscopes and cryo-FLM can also confirm that the axon in one square originates from the soma in a different square.
Thank you, Joe. Our next question, this is our last question, is for Jae. Is bringing lamellas back to the cryo-CLEM viable? And if so, what are the downsides?
Yeah, it is actually a lot of, not a lot, but quite a few labs have success in bringing the lamella back to the cryo-CLEM system to cross-check the milling process because you want to know whether or not you actually mill to where you want to see to define the endpoint. I guess the biggest concern is the potential ice contamination and breaking lamella because of this multiple handling. And right now there are some tools and labs have developed to actually put the fluorescent objective lens into the aqueous chamber, so you can actually mill while fluorescent imaging this process.
Thank you for that answer, Jae. And thank you, Dr. Wright. Do you and your team have any final comments for our audience?
Well, thank you, Jen. Once again, we'd really like to thank our audience, Leica Microsystems, Alvéole Labs, and LabRoots for joining us in this webinar series. I'd like to give a special thanks to Jae, Bryan, and Joe, who really provided all the data and insights into the topics that we've discussed during this series. This closes our three-part webinar series for 2020. These webinars are available to you at LabRoots. And once again, thank you, and we wish you a very happy holidays and a happy new year for 2021.
Thank you again, Dr. Wright, Dr. Yang, Dr. Sibert, and Mr. Kim for your time today and for your important research. We would also like to thank LabRoots and our sponsor, Leica Microsystems and Alvéole, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.